Today, we are going to discuss about evolutionary aspects of plant embryogenesis. A plant embryo is a multicellular structure that contains the earliest forms of a plant's roots, leaves, and stem. Embryo is formed after fertilization when male gamete fuses with the female gamete, and the process by which embryo is formed is called embryogenesis. In animals, the mature embryo usually constitutes a miniature version of the adult organism. In plants, by contrast, embryogenesis generates only a less complex core structure, the seedling, while virtually the entire adult plant morphology is generated by the activities of the apical meristems. Next is evolutionary history of plant embryos. Embryogenesis is a much older process, predating the invention of seeds and plants. The first embryo-producing plants evolved from an algal ancestor around 450 million years ago. Green algae have a dominant haploid phase, and the only diploid phase of their life cycle is the unicellular zygote. After fertilization, a diploid zygote undergoes meiosis and re-establishing the haploid condition. In some cases, zygote becomes dormant and delaying meiosis. In addition, during dormancy, zygotes of many species remain attached to the gametophyte and remain nutritionally dependent on it. So, delayed zygotic meiosis paved the way to the diploid multicellular sporophyte, that is, embryo generation of land plants. You can see here in this image is showing young embryos from different plant groups. This is algae, charophytes, no embryo is there. In the case of bryophytes, here liverworts, hornworts, mosses, these are the embryos. Then in lycophytes, in ferns, in gymnosperms, and in angiosperms, I can see here the upper part of the embryo, that this green color indicates the pro-embryo or apical part of all embryos, where the lower part, that is the gray color, indicates the foot in liverworts, hornworts, mosses, and ferns, and the suspensor in lycophytes, in gymnosperms, and in angiosperms. In the unicellular chlorophytic algae, Chlamydomonas reinhardti, the combination of three amino acid length extension class homeodomain, that is TALE-HT genes, that marks the beginning of the diploid phase of the sexual cycle and initiate zygote development are: first is gamete specific +1, GSP1, that encodes a bell related tail HD proteins. And the second one is gamete specific -1, GSM1, that encodes knotted one like homeobox related tail HD protein. The life cycle of the bryophytes is thought to be intermediate between that of the ancestral green algae and the vascular plants. In bryophytes, similar to the charophytes, that is algae, the life cycle is haploid dominant. Many developmental features are shared between most closely related algal sisters to land plants. One of the major distinctions is the development of a diploid sporophyte embryo. Then, in bryophytes, class 2 KNOX genes, KNOX genes, have sporophyte specific expression, and mutations leads to the development of the gametophytic body plan from diploid sporophytic embryos. After fertilization, the diploid zygote divides via mitosis and develops into multicellular diploid embryo, the sporophyte. Sporophyte produces haploid spores via meiosis, which germinate and develop into new gametophytes. In contrast, The life cycle of vascular plants evolved from haploid dominant to diploid dominant. So, homodomain HD transcription factors have been reported to play a critical role in regulating the sexual developmental program and establishing a haploid to diploid morphological transition during evolution. The inverse process, when the development of a sporophytic body plan in the haploid gametophyte is repressed by the polycomb repressive complex 2, PPPRC2, in psychometrella. The sporophyte is large and contains meristems that fuel post-embryonic growth, where the gametophyte is reduced to a few cells. So, in seed plants, embryogenesis starts with fertilization and ends with seed desiccation and often dormancy. In the liverworts, for example, Mertensia polymorpha, a knockdown of the RWP-RK domain protein leads to overproliferation and sterility of egg cells. This suggests that MPRKD keeps the egg cell quiescent by preventing it from entering mitosis in the absence of fertilization. Thanks for watching. If you like this video, please make sure to subscribe for more. Thank you.